All right, it should be good now. Sorry.
Okay. One of the things that we're missing is the ability to actually visualize these molecules. And so there are certain privileged cases like these CRISPR machines where, oh, I can't see my mouse. All right, I'm going to point to it over here. I know I shouldn't do that. You guys have like a laser pointer? All right, let me see if I can. Okay, I'll just tell you where to look. So on the top right, there's a molecular picture of a CRISPR machine, and can you see that kind of double helixy thing? Okay, that's the RNA wrapping around the DNA. And for some CRISPRs, we can actually visualize what the machines look like. But for the majority of RNAs that we care about in biology and evolution and medicine, we haven't been able to get those kinds of structural pictures. And you know that because you've never actually seen a picture of like an mRNA vaccine, right? And this is one of the key things that we'd love to have. It's AlphaFold has made it possible for us to visualize and include in our engineering pipelines structures, so basically at demand for proteins. And it would be incredible if we had a similar kind of algorithm for looking at RNA molecules. And so my dream for a long time has been to be able to take an RNA sequence that I read about, maybe it's a new virus, or one that I'm trying to design potentially as part of an mRNA vaccine, and to be able to rapidly, in seconds, figure out whether it forms interesting three-dimensional structures like what I'm showing here on the right, and if so, what are those structures? There has been progress on this front over the last 10 or 20 years. How close are we to an AlphaFold for RNA? Okay, so that's going to be the main theme of my talk today. And so on that note, we have some good news and we have some bad news. Okay, so as many of you know, AlphaFold was elicited through a worldwide competition called the CASP trials, the Critical Assessment for Structure Prediction. Okay, and that happened back in 2020. And in the most recent CASP trials, I was invited to help organize and evaluate a new category for CASP, an RNA category. And on the one hand, some of the models were amazing. Okay, so we had 12 targets and we had 40 groups predict. They rose to the challenge of predicting RNA structures. And they produced models like these. If you look in blue, are blind models submitted by CASP predictors for 12 diverse RNA targets of well-defined three-dimensional structure. And in green are the experimentally solved structures of those RNAs, which were released after the CASP competition. Okay, so these are legit blind tests. And for the majority of these targets, the models were visually quite accurate. And in some cases, including in design structures like the four on the very right here, the blind models were closer to cryo-electron microscopy structures than the original design templates that the folks had used that they were trying to make these RNA nanostructures. And so these were quite remarkable and suggested that there was progress being made in the last CASP. And we, like everyone else, assumed that what had happened was someone had developed an AlphaFold clone for RNA or maybe something better and had developed all these methods. But when the predictors were de-anonymized for the CASP conference, we were pleased to see in the abstracts that nearly everyone actually did use deep learning to create their models. Everyone except the top four groups. Those are the groups who provided all the models that I'm showing you here on the slides. In fact, those folks actually intentionally avoided deep learning approaches and used their own expert intuition, as well as classic computational approaches that my lab and other labs had been developing for the prior decade or so. Okay, so we're in an interesting situation where I guess some of us sort of still have jobs as human predictors, but it still kind of sucks because if you want to solve figure out what your RNA looks like, you either have to find one of these top four groups to come and spend a week or two modeling a structure for you, or you have to invest the time and effort to do something like cryo-electron microscopy. You have to do experiments. And that's not going to scale to all of biology or all of biotechnology. Okay, so we really still do want an AlphaFold for RNA. And so what is it going to take to get there? Yeah?
I'm just wondering, I think there are some properties that have like pretty big structure and some of them like more disorder, and I think AlphaFold don't deal with those well. So I wonder
We actually don't know what fraction of RNAs and RNA domains form well-defined three-dimensional structures versus ones that don't. For the CASP competition, it's kind of a gimme. We say, well, we're going to evaluate your target, so we're going to guarantee that these RNAs form well-defined, experimentally resolvable structures. But even that problem is not solved. As I'll return to at the end of this lecture, one of the reasons that I want to get to a predictive understanding of RNA structure is I want to answer your question. I want to go and look inside, for example, the human genome or a new virus and find out, like, is it full of widgets? Are we full of like interesting RNA nanostructures? Does every mRNA in our body have like 3 of these little well-defined domains? Or is it super rare? And nobody knows the answer. If you talk, I've done informal surveys of quote experts or people who've been studying RNA their whole lives. And it ranges from, yeah, like every mRNA will have 3 of these interesting domains, and they're starting like RNA drugging companies because of that belief. And then there are plenty of people who worked in RNA their whole lives, and they're like, yeah, well, maybe, some viruses have 1 or 2 of these domains. So the rate is like 0.01% of RNAs might have these structured domains. And feel free to interrupt with other questions. I don't have that many slides. Yeah. 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 Great. Yeah. So, okay. Yeah. For the Zoom, I'm just going to repeat the last couple of questions. The first question was, how many RNAs? AlphaFold doesn't do well for disordered protein domains. It only does well for the special protein domains that have structure. How many RNAs have well-defined structure? Second question was, how important actually are these structures for function? So the two, there's basically 2, I would say, really interesting modalities for trying to take advantage of RNAs for medicines. Okay, and one of them, there's a whole bunch of people, and you'll hear in the next talk in this series, you'll hear from Stefan Eisman, who I was fortunate to work with too, when he was a grad student at Stanford. And he's part of a company that is trying to solve 3D structures so that we can drug them, so that we can try to design small molecules that will go and inactivate them. And the premise there, it's a cool one. Like, suppose that someone eventually finally does figure out like how Alzheimer's works. I bet you there are going to be like 10 genes that you have to either like 3 of them have to turn on, 4 of them you have to turn off, and a few of them you're going to have to rewire their splicing. And it's going to be highly unlikely that you'll be able to do that by drugging the proteins that those genes code for. But what if all of the mRNAs or the pre-mRNAs that are cut and pasted to form the final mRNAs, what if those all have these kinds of integrate folds? Then these are good targets for small molecules. Whereas the bits of the RNAs that are forming lots of different structures or they're unstructured are unlikely to be good specific targets. So that's one reason that structure, I think a lot of us are interested in structure. But the other reason is that a lot of us are trying to fight fire with fire. So we're trying to, for example, take out an RNA pandemic virus by creating RNA vaccines. And so we want cool functions in the vaccines. And so wouldn't it be amazing if, like, when you got your next, mRNA vaccine for COVID or for flu, that you wouldn't get knocked out. Like the RNA, 1/100th of the amount of the RNA gets injected into you. And that RNA recognizes the cell types that it's going into and says, hey, this is a muscle cell. I shouldn't express myself. I'll destroy myself. But in an immune cell, I'll activate translation. And wouldn't it be great if the RNA does lead to some sort of runaway effect, you could apply a cream with a small molecule in it, like theophylline, and turn off the RNA. So that kind of stuff, I mean, it sounds maybe like science fiction, but natural RNAs have all those functions. And almost all those functions are encoded through special structured RNA elements that we've been able to decipher. This one in particular, this is a structure that my lab and our colleagues actually solved. It was the first RNA enzyme discovered in like 1980. We eventually figured out, it took 40 years to solve it. It turns out cryo-EM is an amazing technique for solving it. It's just this random selfish element that propagates itself in pond scum, in Tetrahymena thermophila. And it's of historical importance because it was this first enzyme discovered. But people are using this and cousins of it to develop what are called circular mRNA therapeutics that appear to be more stable in the human body. They take advantage of the way that this RNA cuts and pastes itself to then cut and paste arbitrary messages to form paste long messages into circles. So I think that's the other reason that often structures do, they tell you about and they let you design really cool functions. Yeah. Thanks for the question. Yeah. I'll have to say for most of this talk, I'll mainly be trying to drive your motivation because I think it's biophysically really interesting. But there's a lot that could happen if we had this power. Yeah. Okay. So what's it going to take? All right. So I think a lot of us feel that there's an issue with data scarcity in the RNA field. So whereas AlphaFold was trained on roughly 100,000 protein structures available in the protein data bank, does anyone know how many RNA structures there are? 2,000. Yeah. Okay. So thousands-ish. Yeah. If you get rid of redundancy, it's probably under 1,000. Okay. So it's about 2 orders of magnitude too small structure-wise. And then some of you who also are familiar with AlphaFold know that it relied on multiple sequence alignments as a core source of data for then predicting structure. Whereas for RNAs, we have only a few thousand deep enough multiple sequence alignments. There's interesting technical and molecular reasons that we can't get deep multiple sequence alignments for RNAs, except for a few privileged molecules. And whatever sequence alignments that we have been getting are typically very, very, they're not very diverse. So they're like, you go to RFAM, your RNA family's website, you can go there now, it's 4,000 families, seems like pretty good. You go to any of the families and like all the sequences are 95% identical. Okay. So there's this data scarcity problem. Okay. So I think what a lot of us have been hoping for is some way to expand the structural data set. And although there have been advances in accelerating experimental techniques like cryo-electron microscopy, some of that's been coming from my lab, we're still, we're able to now solve tens of structures at once for a study, but not hundreds of thousands or millions, okay? And so so my personal goal and my lab's goal for a little over a year now has been to try to find some, way to get lots of structural information to help us train something like an AlphaFold for RNA. And we've had to go back to what looks like a kind of a low information technique. It's been around for more than 40 years. It was actually first proposed at the same time as the first DNA and RNA sequencing techniques were proposed back in like 1980-ish or 1978. And it goes like this. Okay, suppose you have your RNA of interest and it forms these kinds of structures here. It could actually be a pool of a whole bunch of different RNAs too. Okay, there are chemical probes, some of which were discovered 40 years ago, and some of which have been developed and advanced by my lab and others over the last, let's say, 2 to 5 years. And these chemicals, if you mix them into the test tube or into living cells, they'll mark the parts of the RNA that are in loops, that are not in structure. Okay, and then there are a whole bunch of tricks that we and others have developed where you can read out where these marks are sitting on an RNA and put it onto a DNA sequencer. And so from these experiments, you'll get a graph like the one on the bottom where at each nucleotide, you get a readout of how reactive that nucleotide was to the chemical. And if it's reactive, it's probably in a loop. And if it's not reactive, if it's protected, it's in some kind of structure. Okay. And these techniques, because they can accept sort of, it's now possible to chemically synthesize fairly large libraries of DNA templates that you can then in a single reaction convert into a whole bunch of RNA molecules. And because it's possible to use deep sequencing to deconvolve all this information really efficiently, this seems like a way to potentially scale up and get up a lot of structural data. Yeah. And so I went back in the lab a little more than a year ago, so last January, and said, yeah, indeed, we can basically send a text file to a DNA company like Twist or GenScript. They send us back basically arbitrary sequences. And then we figured out how to get these kinds of data like I'm showing on the left. Each row there is a chemical mapping profile for a distinct RNA sequence, and black means shows you the parts of the RNA that are exposed, and white is the parts that are protected. Yeah. And the data are really replicable. Like in a very predictable way, you can say if I invest like 5x more money in my sequencing, I can get that much better signal to noise. And if different experimenters use different sources of DNA or RNA, they get the same answers. That's what this plot is supposed to show. Okay. So, and it's like one-dimensional data, sequence in, data out. This looks like a beautiful problem for for machine learning. Okay. The only thing that's weird about this is that you're not actually like seeing the structure of the RNA. You're getting some sort of weird one-dimensional projection of the structure. So, if you train a machine learning algorithm to be able to predict these chemical mapping profiles for new sequences, like, is it really going to understand structure? Like, and is it going to give us enough information to go and, solve new ones? We going into this actually felt pretty optimistic that indeed such a model, such an oracle would be useful. And here's why. Okay. So, before the age of deep learning, we had to rely on experiments to, solve RNA structures. And my lab was doing these chemical mapping approaches. And we figured out hacks where we could trick the chemical mapping experiments to tell us more than just these one-dimensional projections of structure. Okay, here's one of the tricks we call mutated map. I'm showing you here the chemical mapping profile for a really interesting bacterial RNA that senses adenine. And we had reason to believe it forms an interesting structure. You can see much of the bands here are white, as if they're forming a whole bunch of base pairs. The problem is we don't know if a given band is protected from chemical reactions. What is it protected by? What's it part of? Okay. But here's the idea. What if you just went and you just mutated every nucleotide one at a time, and then remeasure the chemical mapping profile? So I'm showing you here the actual experiments, the data here, and going down the line. Do you see there's this kind of diagonal here? Okay, those are perturbations of the RNA right where we make mutations. Okay, that's kind of a sandy check. But there's also a bunch of other stuff that's off the diagonal. So here I'm showing you, like, if we made a mutation at position 20, we see position 20 pop out of the RNA and become reactive. But also position 40, which is the other red circle, it's also reactive. So then we can infer that those two are connected together in the original structure. Okay, does that make sense? All right. So, and then these kinds of mutated map data, they're really rich. Like, you can actually make out features that correspond to every Watson-Crick-Franklin helix in the structure. You can figure out other interactions that glue together those helices. And we have been routinely using mutated map experiments to go and model RNA secondary structures, which are these Watson-Crick-Franklin helix diagrams of these helical strands. As well as in some favorable cases, we've been making models of these three-dimensional structures, which then agree with, in blind competitions, with the actual structures solved by crystallography or cryo-EM or other classic techniques. In addition, in the modern age, we tend to use this when we're solving cryo-EM structures. We still use mutated map to get the secondary structure and as a cross-check on what we're getting with these other microscopy techniques. So, okay. So imagine that you've got a computational oracle that could give you these kinds of chemical mapping data. Well, we can interrogate that oracle in the same way that we do our actual experiments. And we can see whether it understands RNA structure and also what it thinks an actual RNA, the interactions are inside an RNA. Yeah? Okay. So that's the premise. We believe going into this that if we had an oracle for chemical mapping data, it would necessarily have to understand RNA structure. Okay. So that was the idea. We had the experiment in hand. But we now have to get some sequences to go probe, okay? Because we're not going to get very far if we just probe that adenine ribose switch or a tRNA over and over again, or just things that are 95% identical. Okay, so that was one thing we had to solve. And the other thing we had to solve was, well, how are we going to get the oracle trained? What is the right kind of model to be using? Yeah. Yeah. We don't have very good reagents that mark out helices. So what I'm going to show you is we literally ask a company to make a single mutant here, make a single mutant here, make a single mutant here, just take all the freaking mutants. And then we know that we have all the different sequences. And then I didn't explain this in detail, but when we read out the chemical mapping profiles, we actually use a sequencer to actually know can infer for each sequence what was the sequence and mutations that it had, and then where were the chemical mapping marks as well. We can deconvolve that information. Yeah. Thanks for the question. Yeah.
making mutants, is there any like choice you made, I guess, in terms of like if you have like Mutated, for example?
Yeah.
Is there any decision if you're still requires the opportunity for that?
Yeah. Yeah.
Trained out.
So in the early days when we weren't using high throughput synthesis, we often choose to change a nucleotide to its complement, A to U, U to A, G to C, C to G. Because we saw empirically that that gave us the most information. It was the most likely to make some other partner pop out of the structure. So you can play tricks like that to optimize. You can also, if you have some idea of what structures, you can decide to only make mutations within helices so that you get the most information about, or parts that are protected so you can get the most information about partners. There's also, I went on a 2-year tangent where I worked with my students and I figured out ways to do triple mutations, quadruple mutations, compensatory mutations to suck out even more information out of these experiments. But I won't have time to talk about that detail. It's kind of a fun thing, yeah. Once you have one of these oracles, you can try all these experiments in silico, so it's actually really fun. Yeah.
Yeah, I have a question. Maybe it's a detail is that if you make a mutation at one place, right? What if there are perturbation at multiple places, and what will be the extent?
Yeah, and so those actually occurred here. Yeah, so some of you may have, I didn't highlight, they weren't in red circles, but there's an example. If I make a mutation here, this stuff pops out, but you can see other stuff is popping out too. There's like multiple things that are happening. The explanation turns out that those are cases where the RNAs actually, even RNAs like the sensor, can form multiple structures. Okay, and some of the mutations you make will stable or partially stabilize those alternative structures. So what we're seeing are those alternative patterns. In fact, you can use this method for RNAs whose functions actually depend on forming 2 structures or 3 structures. You can actually dissect those structures using this method. Yeah, they're excited states. Yeah, yeah, yeah. So that's I'm not going to go into that today, but all the stuff I'm going to tell you about offers a window into understanding those kinds of RNA switches. And also, what I'm kind of really excited about, but we'll talk about, is using these approaches, the models that I'm going to talk about, to design novel switches. Like an mRNA vaccine that decides to destroy itself when it's in like the wrong cell type. That's what you need for those kinds of medicines. Okay, so these are the 2 questions that we were left with, last January. We had the chemical mapping stuff worked out. What are we going to do? So we, as you know, most of the really fundamental innovations and developments in deep learning have involved internet scale data sets collected through some kind of like crowdsourcing, bringing together lots of volunteers or maybe not volunteers and bringing together their data through coordinated efforts, like ImageNet for image classification, right? And arguably, AlphaFold is also a beneficiary of crowdsourcing of protein crystallography over 20 or 30 years. That's been organized through the Protein Data Bank. Okay, so it turns out we're fortunate to have crowdsourcing platforms that could help us answer both of these questions. So first, and we have 2 of these platforms. Toward getting interesting sequences that we want to probe, I've led a project called Eterna for the last few years that crowdsources intractable problems in RNA design to people through a mildly addictive video game. And they can send us back these, they have access to a approximate simulation engine, folding engine, and they send us back sequences that fold into structures or have functions that we are trying to create in the lab. And then we and our colleagues at Stanford have actually take their sequences and we make them in a wet lab. We've invented ways to characterize their structure and function. And we return those feedback back to players. And so that virtuous cycle of gameplay and wet lab feedback, this project has now resulted in over 30 scientific publications. Many of them are actually co-authored or in some cases lead authored by these video game players who in some cases have never been to college and have no expert scientific background. And so this community, we engaged them last year to go and help us find a bunch of sequences that they thought might fold into intricate, never previously seen RNA structures. So that's where we got our sequences. And then in terms of where we get models, well, I have wonderful people in my group who develop machine learning models and great collaborators. But a reason that you could do better if you just had lots of ideas. And so we turned to this platform. Kaggle. Who has heard of Kaggle? Okay, it's a thing, right? Okay, you know what it is. Okay, like for bio audiences, I have to explain to them. And so as you all know, Kaggle has a really rich community of folks who teach each other how to, do data science and how to create awesome models. It's pretty friendly, but there's this nice driving force where people are also competing for prizes. I first started working with Kaggle 4 years ago during the pandemic, and we brought together Eterna and Kaggle to solve a problem that we had encountered in our work on vaccine research, where we were trying to figure out principles to stabilize RNA vaccines through structure and through sequence. And we had come up with really cool models that actually form the basis of, like companies and things. We got even better models for mRNA degradation through this open vaccine initiative and through a Kaggle competition whose training and test data came from the Eterna platform. Okay, so that was the precedent, this dual crowdsourcing approach that we then brought to bear again last year. So the new competition I'm going to tell you about is called, it involves roughly 1,000-fold more data than the open vaccine competition. And it's seeking to not just get a model of RNA degradation, but basically general models of RNA structure. Okay. And this competition, the data set in this competition, we decided to call it Ribonanza. This is the banner from the Kaggle competition, and we launched it last September. Okay. And at the beginning of the competition, we had data on a little over 1 million RNA sequences for the RNA aficionados. I gather there's some of you here. We used the shape and DMS chemicals. We derived these from various sources, but the plurality, the biggest source, was the Eterna platform. And then we closed this competition in December, and we scored people's models on data on another 1 million RNA sequences. And to help ensure rigor and to ensure that we would have fun as hosts, we decided to not collect the data until like near the end of the competition. So that way, for the first 2 months, we were all in it together and trading a bunch of ideas on, on the forums and like, hey, have you looked at this particular test molecule? Here's what mutated map is. You can use it as a sanity check for your orbitals. And so that was really fun, but it also made sure that there was just no chance of like leakage of our test data to influence people's models. Okay. And we also implemented other safeguards to ensure generality and rigor, one of them which I'll mention next. Okay. So we closed the competition last December, and the first thing that happened was a bunch of the Kagglers were like, "Hey, something looks totally wrong. I was supposed to be in like 50th place, but I'm actually, 150th place." There was what's called a shakeup. Those of you who are taking part in Kaggle, this is like super scary and annoying, right? Because you thought you were at a certain place in the competition, then you get worse. We were very pleased when this happened because we intentionally wanted there to be wanted folks the so-called private leaderboard, which are test data that no one has seen. We wanted to make sure that at least for the competitors who weren't paying attention, they do worse in the private leaderboard than they did on the public leaderboard, which is a continuous evaluation test set where Kagglers can't see the data itself, but they can get scores about this continuous evaluation. Okay. Does anyone guess what we did that was different between the public and private leaderboard to ensure generality? And
what's the x-axis?
All right, the x-axis is a ranking of someone. So competition is like going on, but it hasn't closed yet. So there's a continuous evaluation and a leaderboard where you are relative to everyone else. Okay. But the actual prizes, who wins, is governed by something called a private leaderboard, which actually we didn't have at the beginning of the competition. But those are test data that no one is able to see or get scores for. Yeah? And that's what determines the prizes in the best models. Anyone guess what was the difference between the I'll look in front of me. There are different data sets for one thing. What?
You just keep a family.
Did I skip a family? That was better than that. Yeah. If you use like a vanilla transform model, what is it? What is it fail? What is the most notorious thing it'll fail at?
Long context.
Long context lengths. Yeah, length, right? Okay. So we intentionally, and everyone knew this because we told them what the private network sequences are going to be. It's not the answers. This is a meme that they had published during the competition. The trained sequences, the test sequences. So we intentionally made it so that all the test sequences had a wildly different length distribution than the trained sequences. Okay. And so, and this is just a trick that we learned in the open vaccine, the prior Kaggle competition as well, to help enforce generality. And the top competitors obviously paid attention to this, in fact, and they post these memes. And if I zoom in on the top competitors, so lower is better in this ranking, there actually is pretty good agreement. Like if you're in the top 10 in the public leaderboard, like before the competition goes, you'd have a 70% chance of being in the top 10 in the public leaderboard. Okay. So that was cool to see. The other thing that we noticed just looking at the scores was that there's a rule in the Kaggle competitions where the teams, after a certain period, for most of the competitions, they can share everything. But there's a rule that you shouldn't be sharing things for the last week of the competition. Okay, just to ensure we get independent models. And what was cool is if we blended the top 3 models, we actually got a better submission. Lower is better here. Okay, suggesting that folks were doing something that's slightly, maybe they were similar, but they were doing something slightly independent. In addition, we had our own baseline model. So I had recruited there's a Kaggle grandmaster named Rhiju, who started in Kaggle because of the prior open vaccine competition. Actually, he's a graduate student. And then we recruited him to come and help us host the competition, sort of to be on the other side and develop a baseline model. And it was very pleasing to Rhiju and to me and everybody else that, again, the Kagglers outperformed what we were trying to do on our own. I'll tell you more about that RNA deformer model in a second. Okay, so that's the overall performance. What are these models? These are the names of the models. Our baseline is RNA deformer. The top 3 models had, when described by their authors, were a squeeze former with BPP continuity intention, a transformer model, an evil former style alpha model. As you maybe might have guessed, these are all transformer style models. Okay, but they're not vanilla transformer models. Okay, so I thought I don't usually get to like geek out over architecture, so I thought I need to it in this audience. I'm showing you here RNA deformer on the left and 2 of the top Kaggle models on the middle on the right. The red boxes are to highlight that each of these models has a classic transformer encoder. So multi-headed self-attention in the gray box and the feed-forward network right below it. Okay, and these transformers are operating on the residues, the nucleotides of the RNA. Yeah. All right. Does that make sense? Okay. Okay. But okay, they're not vanilla. The vanilla transformers were all like in 200 place, 300 place. Okay. What these top models all did is they were independent of each other, but they all did a couple things that we weren't necessarily expecting. Okay. One was they all had a track where they're processing a pairwise representation explicitly of the RNA. Okay. Multi-head attention produces an implicit pairwise representation, but these are all processing pairwise representation explicitly. Those of you who know the details of the alpha-fold architecture for proteins or the Rosetta-fold architecture for protein modeling know that those also have explicit pairwise tracks. And these pairwise tracks are used to bias the attention. Okay, that was a feature in all of the top 10 models actually. In addition, I wasn't expecting this. The top 10 models all, in addition to classic transformer operations, they all had convolutional anomaly blocks as well, which I thought you would be able to get rid of, by this point. But somehow, like picking out little localized sequence motifs is just best done with a convolutional operation, and so that is preserved. Although I like it in different locations in these different independent models. Okay. They also all use relative positional encodings, but totally different relative positional encoding. I'm getting into the weeds here for the aficionados, but I thought you'd be interested. They all use different relative positional encodings, like literally the top 10 models. One of them used rope, one of them used like dynamic positional encoding, another one used like they just put in a pairwise representation of the distance matrices like, or the sequences to matrices, all different. Okay, so we have all these models. Did they understand RNA structure? Right, that's what I cared about the most. They did well in this leaderboard, but does that mean they understand any structure? Oh, I also want to point out one thing that I also was surprised by. So none of these models, although they had bits and pieces that resembled AlphaFold or other models, other kinds of models, none of them actually took into account the fact that RNAs are molecules that fold in 3 dimensions. Like, I would think that would be the most basic inductive bias you want to put into a model, but none of these models include that. Any 3D kind of simulation. Yeah.
Could you explain the pairwise, like, whether
Yeah, sure. So, okay. And also, well, let me maybe make another point that I found interesting. Okay. So the way the models work, I'm only showing you the core block here. They process sequence representations, which is a bunch of features for each position, each residue. Okay, like 512 features for each sequence. Okay. But they also process a pairwise representation, and you can think of the pairwise representation as like one of the pairwise representations could be which nucleotides are likely to form Watson-Crick-Franklin pairs with other nucleotides. Yeah, that's the kind of thing that's kind of natural to be processing. And in fact, when we looked at some of these pairwise representations for different models, we actually see like features that look like they're trying to simulate Watson-Crick-Franklin pairs. Yeah, and some of them are processing like what might be triplex interactions. Some of the pairwise features are comparing the strengths of different potential Watson-Crick-Franklin stems. Yeah, so that's the kind of stuff that can get built up in a pairwise representation. Does that answer your question? Or what?
it's just
Could
like
be
the
in there.
relationship between 2 different nucleotides?
Yeah. 2 different nucleotides in the RNA sequence. Yeah, 2 different positions. Yeah.
So none of the 800 even had like something like triangle attention or no 3D.
So in this third place model, and only in this third place model, one of the 2 models that Dan Kirstev submitted was involved a triangle update. And that was the only one. So I'll actually return to that. Thanks for asking. Yeah.
Do these models use like dynamic programming based algorithms to sort of like bringing in everything?
Yeah. So it turns out all the top 10 models at some point use the outputs of classic dynamic programming methods. Nearly everyone converged on using a method called Eterna, which was has an interesting history because it was a classic kind of method. It was called contrafold. It was developed at Stanford 15, 20 years ago. And then my group and the Eterna project recalibrated all the parameters based on prior Eterna data sets. And so that Eterna fold ended up being what almost everyone used. Actually, what everyone, amongst the top 6 models, all top 6 models use those representations. But as I'll show you, you can actually translate them for you. You don't have to use them.
So I'm assuming that pair representations derived from like that you did.
No, so the pair representations, the way that they're initialized, actually for most of these models, the way they were initialized is through a predictive base pair probability matrix that came from one of these classic algorithms. So that was the initialization for most of these. For most of these, remember, they don't have to make a prediction on a new test sequence. We don't have the experimental mutation method.
So this would just be mostly like
Yeah, that's an example of one way you could initialize.
I see. And are they all consistently the same, or each team sort of tweaks it a little bit different?
Each team made different choices about when and where to apply a convolutional
I mean,
operations.
how they initialized it.
Different teams use slightly different choices. So as an example, Jujun used base pair probability matrices, that's what this BPPM means, but also added on to this a matrix that just encoded the sequence distance, like between any pairs of residues. Like if sequences are, this pairs I and J are 10 apart, so it gets like a 10. You know what I'm saying? Yeah. And there's a similar kind of encoding that AlphaFold uses also in its pairwise distributions. Yeah.
I know you mentioned that there's like the scarcity RNA structural data sets.
Yeah.
Do you have a sense of whether that data set includes modified RNAs?
Yeah, good question. Yeah. So right now we are just looking at unmodified RNAs. If you can remind me, the question again near the end, I'd love to sort of speculate on what we think is modified RNAs. I think we can do a lot. It's just a matter of we're just doing. These are all unmodified though, just A, G, C, and U. Okay. Okay. So did these models learn RNA structure? So the way that we're going to actually assess that is through in silico mutated map experiments. Okay, so this is coming back to the mutated map idea. What I'm going to show you here are computational predictions from these models for an RNA that I think is interesting. It's a little widget in coronaviruses that tricks ribosomes into pausing and then occasionally slipping back by one nucleotide and producing different proteins. Okay, and it's in every coronavirus, including SARS-CoV-1, SARS-CoV-2. Our lab and others have solved this in SARS. In the SARS virus has been known to solve this element structure in the MERS virus. Okay, so here are mutated map predictions, okay, not the experiments from one of these non-vanilla transformer models. And you can see that it thinks that there's some sort of interesting RNA structure there. I can't point. Okay. Like if I make a mutation at position 45, I get something happening out here at position 100. So the model thinks that there's some connection between those pieces of the RNA. It's a little bit easier to see if I subtract out the predicted reactivity for the wild type molecule and just show you what shows up in the mutants. And you can see there are these like blue splotches. And there's 6 of those, and those correspond to each side of 3 potential stems that this RNA might form. So that's what this model is thinking, and if you can think, that this RNA has 3 stems. And actually, all the top models in Kaggle made the same predictions for this MERS coronavirus simulation with these 6 features corresponding to them, inferring there are 3 stems in the RNA. And so that was cool to see that convergence, but of course, are they right? You have to do the experiments to see if they're right. So here's the experimental data, which we included in the test set, and they're dead on. Like within experimental error, these are agreeing. And it's not just these stem features, but there's other features out here that are kind of delocalized. And I got a question earlier about like, what does that mean when there's other stuff happening? So these are alternative states, excited states of the RNA. And the experiments see them, and the models also predict them. Like they understand alternative state structures too. So I thought, yeah, this is really cool. And you know, what does this structure look like from the experiments and from all these models? It looks like this is a 3-lobe stem. And this actually appears homologous to what exists in SARS-CoV-1 and SARS-CoV-2. Okay, so that's another kind of sanity check for this particular case.
Professor Das?
Yes.
Are there intractable modifier-based mapping you're doing here capturing transient long-distance interaction as well, or is it just specific contextual, like kind of stable stat?
To a pretty good approximation, the chemical mapping approaches show the mean chemical reactivity of an RNA nucleotide summed over all possible structure, over all the structures that it populates, weighted by their actual weights. Okay, so if something's protected 95% of the time, then you see like 95% protected. Does that make sense? Okay, so if there's an excited state that's around like 10% of the time, it's neither upweighted or ignored in the chemical mapping profiles. But you want to get a really good picture of that excited state, you have to stabilize it, which happens sometimes with mutants that we're making randomly, or we have a kind of a scheme for trying to stabilize those excited states because we care about them for some other RNAs. Within this data set, I don't know if we included a whole bunch of RNAs that were switches that have alternative or excited states. But I'm getting so many questions about it that I'm now seeing that I should go back and actually pull out those examples and have some slides on it. Yeah, these are good questions. Okay. Okay, so now we have like these pretty interesting models. They look like they're passing some sanity checks. They seem to be understanding RNA structure. and not just picking up on spurious correlations. We actually have too many models. Like it's an embarrassment of riches. And any of you who've taken part in Kaggle, you may know there's this dirty secret that to win at Kaggle, you not only have to have like really good models, but you have to like, you typically will blend like a whole bunch of different models. Okay. And this is super annoying, right? For people who want to use your models later, because they have to run your code with like 20 different, I've literally the first place model here averaged 27 predictions with their model with different parameter settings, and like they forgot what some of the settings were. Like it's super annoying. Okay. Because they're trying to win this competition. Okay. So, so one thing that we did, which I don't see happening that often on Kaggle, but it's kind of a no-brainer, is we just decided to integrate everything from all the different all the compelling features of all the models into a single model. We did that two ways. One way was we just took the features that we thought were most interesting in the models. And when I say we, I actually mean all the top Kagglers. We're on like all in a Google call together. We're like, hey, what's going to have legs here? And we all kind of made an agreement that triangular updates is something that we wish we had all explored. Third place model, they looked into it, and then they're like, okay, so let's try that. And we'll go use our compute to go and retrain the model. And similarly, I was actually annoyed that folks were using these conventional Eterna fold and these old school dynamic programming models for RNA secondary structure. Okay. And so I was like, can we just ditch that? And we just started de novo. Okay. And because it turns out, again, third place model that included these triangle updates and a few other tricks found that at least half of their predictions did not rely on having to use those classic models. So that was all like kind of eye-opening. We're like, okay, let's see if we can make a unified model. The other trick that we used was we were like, we have all these predictions from all these models. We actually asked people to make predictions on that million test sequences. And we reasoned that we could suck out some of the information that they had learned about RNA structure by using their predictions as pseudo-labels for training a single final model. Okay? So I don't know if you know, like in Kaggle, this is the other trick that people use. They take predictions from all different models, they average them or glue them together, and then they use that to predict, you know, their next generation of models. This is sometimes called like noisy student learning. It's called pseudo-labels, and it has other names too. Okay, so we took all the predictions of these just models and all the interesting features, created a new network called RiboNanzaNet, and I'm showing you the performance of RiboNanzaNet. Here, lower is better again on the two different leaderboards, so it's doing better than the final one that uses all of these pseudo labels is doing better than any of the top 10 previous models. Okay, so that was very good. Okay, and now we have this cool thing that maybe we can use to do science. Okay, so I'm going to tell you a couple of applications, and then I'm going to like maybe describe an open problem that I think you might find interesting, and then that'll be the end of the talk. So we have now taken RiboNanzaNet, and it's doing a silly thing. It's predicting chemical mapping profiles, which no one really cares about. But we've been fine-tuning the model to go and like predict things that we and others care about. So I'll show you a couple of examples. Okay. So one example is we went back to this open vaccine problem, trying to predict the degradation rates of different RNA sequences. Okay. And so, you know, we had our training set and the test set from that last Kaggle competition. We took our RiboNanzaNet model and said, hey, try to predict these these degradation data instead. What do you get? Okay, so here I'm showing you, again, lower is better. In black are all the prior open vaccine models trained on roughly 3,000 profiles, on 3,000 sequences that we collected in 2020 during the pandemic. And here's RiboNanzaNet now trained on a much bigger data set, different kinds of chemical mapping data, but fine-tuned to the same training data here. And it's totally crushing all of these prior models. It's even beating this RNA deformer model, which is a post-competition model that Rhiju, our collaborator, this Kaggle grandmaster, had prepared. And that averaged 50 models and used a whole bunch of tricks. So here, you know, and it literally takes, I think, 10 minutes to do the fine-tuning on a Kaggle notebook, super fast, and then boom, you know, we're winning the competition. Okay, and it also, like, the predictions it makes, like, they make physical sense. They catch like all these discrepancies that the prior models didn't catch. So as an example, here's one of the little RNA fragments that we probed in open vaccine. These are data on which parts of the nucleus are hotspots for hydrolysis. Okay, this is the kind of thing. This is the kind of thing that prevents like an RNA vaccine from getting shipped at room temperature. Okay, or using it to freeze. Okay, for this particular molecule, the previous open vaccine Kaggle models, they didn't predict it right. They predicted this RNA would be white or frozen or not. They wouldn't have these hydrolysis hotspots. Okay, but you know, like this fine-tuned ribonucleic model, it knows that there's stuff that's going to be cut out. You know, and you go and I'm not showing you the it's in the preprint that just came out, but we also applied the carbon-antinet model to a bunch of full-length mRNA degradation rates that we collected with an independent measurement, and it totally outperforms these prior Kaggle models. So it really generalizes. I also, let's go back to the beginning of the talk where I wanted to be able to model new RNA structures, you know, and do these three-dimensional structures. So as I've already kind of alluded to, just from this kind of mutated map analysis, we can trick one of these models into telling us what it thinks the structure of the RNA is. Although it's kind of indirect, right? I'm like, yeah, look at these blue splotches, and of course, this and that. So we went ahead and instead said, well, let's just, you know, let's just fine-tune the model to give us directly the set of base pairs that it thinks are in the RNA. Okay, rather than asking it for the its predictions on all these mutants. So we did that, called ribonucleic SS. For this RNA, it predicts these three stems, actually with a slight preference for a fourth. And then, on that case, it just passed the sanity check. It predicted what we expected to predict. But then we've been applying this now to RNAs where that have interesting three-dimensional structure, including these last Cas targets. Okay? This is how we started the talk, in that last blind competition. We had noticed, and as well as the folks who were applying deep learning there and had sort of failed to make the best models, that their models often had the incorrect secondary structures, incorrect pairings of C and G and U and A. Okay? And if you get that wrong, you can't figure out how these helices are going to dock together in three dimensions. Okay? And they use algorithms like spot RNA, another deep learning algorithm. And I'm showing you here the prediction of spot RNA for one of these Cas targets. This is it doesn't matter what it is, but it's a ribozyme. It's actually a ribozyme in your bodies that processes an mRNA. This is what you get from spot RNA. This is what you get from ribonucleic fine-tuned for secondary structure. They're quite different, yeah. This one happens to be more accurate. What's really cool is if you then feed this secondary structure into a model called TR Rosetta RNA, which was the best. It was the best of the worst. It was the best deep learning algorithm in Cas15. Then, whereas in Cas, it did a pretty terrible job of predicting the structure. Experimental structures in white, blue is the model. If you just give this same algorithm the secondary structure coming out of ribonucleic, things just click into place. Okay, so this looks like it could be really useful. You know, just immediately as a slot in, you're just getting the secondary structure right for these 3D models. But not everything's perfect. So here's another Cas target. It was designed to form this like beautiful triangle shape by Ebbe Andersen in his lab in Denmark. Here's what you get with that TR Rosetta RNA algorithm that I described previously. It kind of squashes this equilateral triangle into some other weird shape. And then when you feed in the ribonucleic information, it still squashes the RNA. So we're in this interesting situation where it looks like there's some like interesting information in ribonucleic. It's doing a really good job with secondary structure, but we may not be at the scale yet, or we haven't developed the right model yet to get the information we need to model three-dimensional structure.
I might miss it, but how did you get this 3D? You based on the second from you feel.
TR Rosetta already accepts if it allows you to input, I guess, the secondary structure to guide its modeling. And so we went ahead and took advantage of that.
Purely based on secondary structure.
Yeah. It uses that, it uses multiple sequence alignments. Yeah, and then it's a neural network that actually produces a histogram, and then it feeds that into Rosetta.
Is it possible that probably maybe that's a fundamental, maybe secondary structure also has a fundamental issue?
Yeah, I think so. It's like a bottleneck that we probably don't want to have.
Yeah.
Yeah. So that's exactly the next slide. So this is like an invitation to all of you. You want to work on a really, really interesting problem with like world-changing implications. this could be the problem, and this is the right time to work on it. Okay, so RNA 3D modeling remains no problem. Okay, as of last week, we have this large data set, and we have a really interesting set of models that are trained on those data, but none of those models have tested whether this inductive bias, like the most basic inductive bias, of modeling the RNAs as a bunch of three-dimensional chains. Why? Partly because it's computationally expensive, and so in Kaggle, this is one of the issues with using Kaggle, that people just want to they want to win the leaderboard and they need to iterate really fast, and so they won't say, well, I'm going to invest the next 6 weeks just going for a single architecture with 3D modeling and trying to train. Okay, they'd rather say, well, let me just hack the transformer in a million different ways and see if I can drive my score better. Okay, so that's like a missed opportunity. Okay, so that's number one. And what I think is great about this, so the data are available at the scale of millions of sequences. We've also published the solution data, which means that if you want to, you can use those for training. We have 2 million sequences. But better, you can evaluate your model. So if you have your model, even if it simulates three-dimensional changes, if it predicts chemical mapping profiles at the end, you can see whether you're doing better than 800 Kaggle teams. Yeah. And the solution files are public as of last week. So it's ready. It's well defined. It is a retrospective analysis, though. Okay. So sometimes if you already have if you have the data, it's available. You can beat the state of the art for silly reasons. But that's the other cool thing about it right now. There's more data coming, and there's prospective evaluation starting in April. So the next cast is starting in 2 months, end of April, okay, less than 2 months. I'm organizing it, which means I can't predict. And I'll be assessing it. So I also can't help you, although I can guide you to data sets and to potential, modeling methods that you might find useful. But this is great. So it's, I think it's possible that in this cast, RNA modeling will get solved. Okay. Yeah, it'd be great if one of you did that. So, so that's oh yeah. So my contribution, I'll be organizing the CASP RNA category. We're also collecting data, including some of the RNAs that are in RiboNanza. We're now getting their three-dimensional structure to crowd it. So I'm intentionally not showing you the structure we're getting. I'm just showing you these raw data, they're called 2D class averages, to show you that we are getting the data, because I want to keep them blinded for the CASP competition. So that's going to be there as a rigorous test. In addition, and this one I just invite you to do if you don't have to be part of CASP. So we're taking the current RiboNanza and secondary structure models and just scanning genomes, human genomes, squid, octopus, mouse, E. coli. We're finding that the models are predicting that there's interesting, they're called pseudoknots, that are common throughout RNA genomes. I mean, these obviously predictions will be wrong, but the cool thing is we can test them experimentally. So we're scaling up our experimental pipeline from 1 million to 100 million sequences. We are hoping to get to 1 billion sequences by the end of 2024 or beginning of 2025. But yeah, if you develop a 3D modeling method and you're willing to go and scan some genomes, if you send me a list of 1 million sequences, we'll just include it, we'll synthesize it, we'll give you back the data, and we'll use it to train the next model. Okay, so that's happening, and that's just fascinating, both in terms of training and testing machine learning, but also just for biology. This is going to be the first time that we're going to have maybe an atlas of all the complex structures in viruses, humans, squids, octopi, etc. Okay, so quick summary. There's no AlphaFold for RNA. We learned that in the last cast. Actually, all the stuff I've shown you here, it's this funny thing where we just love these competitions. They've been driving progress, and they've let us evaluate progress in RNA folding. The CASP competitions have revealed that there's no AlphaFold for RNA yet. They've motivated us to develop chemical mapping approaches that can be scaled up. And that's led to this RiboNanza effort, which itself involves 2 other competitions, one on Eterna to solicit the most interesting and complex RNA sequences and structures, and another one in Kaggle to train and test models based on those data. Result of this effort, we just put out the preprint, are a bunch of transformers, non-vanilla transformers. They outperform prior models, including most of my group. They appear to be learning representations of RNA structure that are accurate, and they can lead to better 3D predictions, but there's still work to do. There's still like this last step that we haven't done of putting 3D modeling in as an inductive bias to these models. And then, we're really hoping that everyone in the community and new people, maybe people like you, are interested in these data sets and joining pretty quickly because the next CAP is happening April 2024, and those will allow for a really rigorous readout of whether the problem is solved.
For CAS 15, you
Yeah.
said that top performing models that come from, I guess, domain experts that
Yeah.
introduce inductive biases.
Yeah.
It does include three-dimensional.
Yeah.
Have you guys tried just literally just adding that to run an answer that?
Yeah, so the inductive bias in the 3D modeling is this. Okay, I mean, there's so I'm a human being. If I was participating in CAS, the way I'd win is I'd go run several different secondary structure prediction algorithms, and I use my prior knowledge of which ones are more accurate for which kinds of molecules to pick a secondary structure. In some cases, I could guess that the target is coming from a human group designing a triangle. Like there are people who do that, right? They're like, oh, like okay, there's some other human being who's giving me this problem, and then they're like, okay, let me make sure that my final structures look like triangles. You see, okay, so there's humans doing all sorts of deep learning stuff, but it's biological deep learning, right? People like pre-trained over the last 15 years. Yeah, it's been very hard to automate what, at least for me, to automate what I've been doing or what other groups have been doing in these competitions. I do think that well-curated data set is still, unfortunately, is going to be the way to go.
What do you view as the grand challenges ahead of kind of going 5 or 10 years into the future after you develop the comfortable for RNA? How do you then translate that into the next generation of like drug development relevant kind of RNA structure?
Okay, there's kind of the obvious next steps, which are the most interesting RNAs in your body that are in medicines like CRISPR's are actually RNA protein machines. And actually, many of them also operate in DNA. Okay, so there's a real, if there's a scarcity of data on RNAs, and there's an even worse scarcity for RNA protein interactions. Okay. So that's like obvious next steps. Okay, I don't know if any of you saw that there was a blog post from Isomorphic Labs in DeepMind in last fall. They say, hey, we updated the AlphaFold that can model RNA alone and RNA protein complexes. But and they have some showcase examples. But then if you look at the bar graphs of their performance, they're just abysmal. Like the LVDTs are like 20 out of 100 on average. They're worse than they ran the CAS targets, and they're worse than the human CAS predictors, right? So and they're DeepMind. They should be able to do better. But I think they're probably the best that you can do with existing data. Okay, so I think those are kind of obvious next steps, RNA protein machine, RNA protein. Okay, but I think much more exciting than that are in design. So it turns out even the problem of designing RNA that has a target secondary structure is unsolved. The benchmark for this is called the Eterna 100. It came out of Eterna, the video game. And there's still no one that can solve all 100 problems in the Eterna 100. It's in silico, they test that. It's like, yeah, so that's, I think, shocking. And then design of RNAs that have multiple states or RNAs that have all the interesting new 3-dimensional structures is also basically a wide open area. Okay. The thing that I'm most interested in is that most of these machines actually dissipate energy, and they act like little computers. Like there's this thing inside your body called the spliceosome, which, like in different cell types, will take each of the RNA messages that's made in your nucleus, in the cell nucleus, and then decide how to cut and paste it into the appropriate message for that cell. It's like this remarkable computation. The spliceosome is derived from a pure RNA machine called the loop intron, and its heart is still based on RNA. And the way it's making the computations appears to be encoded by how the mRNA it's processing is responding to other proteins in the cell. And the machine is using energy, and it's doing proofreading, and it's got like 100 states in it. And I don't even know what the models can look like for being able to take a set of RNAs, proteins, and DNAs, and being able to make a movie of it, as opposed to just the list of the states, or in this case, the dominant state. So that problem of modeling and designing kinetic machines that dissipate energy and actually act as computers, I think that's the end of molecular biology. And that's where we want to be, like after this. Going closer to the end.
So it seems like for a lot of these competitions, you're kind of treating the cryo-EM structures as ground truth.
Yeah.
Are you kind of worried about that, just given that some cytoplasm, you're less dynamic?
We do, yeah, we do. So Rachel, actually, I should just quickly flash the acknowledgements here. These are the people who've been doing the work with me, and Rachel has been leading the cryo-EM efforts. In the last CAP, Rachel kind of co-led the assessment with me, and for all the cryo-EM targets, she also assessed all the incoming models by directly comparing them to the experimental data, which I think is really cool. So there's less kind of model bias. Like maybe our inference for the structure or structural ensemble of the RNA is wrong, but we just directly compare it with data. So I think that's kind of thing is important. What you're talking about, because RNAs actually have dynamics. Yeah, did I answer your question? Yeah. Okay.
By data, do you mean like the images or like the density preference?
Okay, so what we did last time was density, the final density maps. But what we're hoping to do in this next cast is to have at least a few categories of RNAs that we know have interesting motions. We'll accept, our hope is to then ask for people to not just to give us 5 separate structures, but to give us 5,000 number ensembles of what they think the ensemble, the cryo ensemble of the molecules. And we'll directly compare those to the micrograph images, the separate molecules.
First of all, thank you for your talk. I think I read your first today. Very excited.
Oh, good.
I bet it costs a lot of money to get all that extra data.
Yeah.
And turn it down. The rest of the community. Thank you for that. I have 2 questions. One is, have you done any studies on how well your predictor works on synthetic versus naturally occurring RNA?
Yeah.
And the second one is about just cast competition. I know previously RNA puzzles had like basically expert mode where like people make submissions and they have more time, and then there's like size. Sorry, it's just looking at.
CASP has been like that too. Actually, the RNA puzzles was inspired by CASP, but they have like a 2-day server prediction, and then the server models are made available actually to everybody, and then humans have typically 2 weeks. RNA puzzles is like that too. Yeah. So I think that that'll continue in the next CASP. Okay. Going back to the economics, turns out the costs of synthesis and sequencing have been going down dramatically in the last 2 or 3 years. And so originally, when I was like a year ago, or a little more than a year ago, I thought we would only be able to get data on maybe 10,000 to 100,000 sequences. That's how much my academic lab would be able to afford. But then we scaled up to 1 million within a few months, just because there's a really nice competition going on right now for making like a $100 genome or a sub-$100 genome. We're benefiting from that. And similarly, synthesis folks, there's strong demand for their DNA libraries. And it's going to get stronger with all the bio AI startups that you're all hearing about in this series, in the seminar series. And so that's going to drive down the cost there, and increase the scale of the libraries. So actually, that's probably why I said we're going to be doing 100 million sequences this year. We can do that, budgeted. But that's why I think we can get to 1 billion next year. Not because I can't actually afford that right now, as an academic, at least as an academic lab. But given the way the trends are going, we will do it. Synthetic versus natural, I haven't. It's hard for us to make that call with the ribonanza. We have a bunch of synthetic 3D nanostructures that are part of our advanced competition. They turned out to be easy to predict. That's probably because we designed them to form really well-defined secondary structures that were easy to predict. So I haven't done the detailed analysis that we want.
I have a quick question from the Zoom.
Yeah.
So, Piazza is asking,
How does the Eterna crowdsourcing work? Is the mutation data that you put in the game and the putative structures from Eterna are experimentally tested?
The way that the Eterna works, well, I invite you to play it. It's at eternagame.org. You can also download an app on your iOS or Android. The game, when you play the game, you don't have access to experiments. You have access to folding engines. You will soon have access to RiboNAND, for example. But once you've gone through the first round of a puzzle, and you got the experimental feedback, will be made available to you in game. So you can see it for the designs you've already made. But because the game is like making new sequences and new designs, you don't get feedback on them. You don't get experimental feedback right away. You get some sort of simulation.
I have one more question. So you mentioned that you use some degree of synthetic data from like, I think ensembling a bunch of different secondary structure models.
Yeah.
Did you experiment with like different ratios of synthetic data to like rapture data? I know, with AlphaFold 2, they tried like different relations there to get to the right balance.
Yeah, Truman did quite a bit. What we found is that it's best to start training on lots and lots of pseudo labels, and then you anneal on real data at the end. But all the real data you have at the end. Yeah. So we tried to I don't know, this is all like black magic to me, but and I can wave my hands and say, oh yeah, that makes total sense, because you don't want to have any crappy pseudo data in there, of the training, but I don't know. That's just
Could you speak a little bit more to the Kaggle shakeup
Yeah.
that you're talking about?
Yeah.
Like basically, because I think you were mentioning that it was largely shape and DNS data that was informing the public data set.
Yeah.
And then all of a sudden there's a longer set of sequences.
The longer, yeah.
that matter.
Yeah.
Is that from how you guys are running experiments, or like, can you like describe what was going on there?
We intentionally picked longer sequences, and we gave those sequences to everybody and said, we need your predictions. Your final scores will be based on this private leaderboard, these private sequences, or these test sequences. So they knew that there were going to be longer sequences, but it's just that a lot of folks, they didn't do the internal sanity checks. They used absolute positional encoding, if they're doing transformers. And so they got susceptible to this big shakeup.
So is this like 100 mer versus 200?
Oh, yeah.
500?
They were, let's say, roughly 100 MERS, and then they went up to, just like 130 MERS. Although
It's only like a little bit.
it's only a little bit, for most of the sequences. But there were a few sequences in the test set that were like 200, 500 MERS, 500 MERS. There weren't as many. We couldn't afford to make as many of those.
You talked a little bit about this.
Yeah.
Has there been any notion of using function? I'm actually not sure, like, from an absolute standpoint, what proportion of RNA has some sort of functional
Yeah.
or
Yeah.
like using function biostructure.
Yeah, so a very, what's very top of mind right now in RNA medicine is designing RNAs that will produce more proteins per RNA molecule, mRNAs, for mRNA vaccines and therapeutics, because that would allow, my kids to get injected with 10x less or 100x less RNA, and so they'd be much less likely to get knocked out with runaway immune effects. And so they're the function of the RNA is like it's an RNA message. Its function is literally to get printed into proteins. And so there's been a little work on taking some like foundation model type approaches with existing natural RNA sequences. There's a paper from actually Sanofi from about a month ago or 2 months ago, preprint, where they tried to fine tune towards predicting protein expression. And it looks like there's some signs of life there. I don't think that is, nailed yet.
My second question is around, so all of this like prediction of like post-splice. Has there been splice in RNA?
Yeah, and in fact, I would say that a lot of the work on in industry is actually focused on pre-mRNA splicing. Actually, I have an expert right here. So companies like Deep Genomics very early on pivoted to trying to design antisense oligonucleotides, so little snippets of DNA or stuff like that, that would go and rewiring or reconfigure splicing pathways to replace proteins. And there are famous examples, recent examples of therapeutics based on rewiring splicing through those kinds of antisense oligos and small molecules. So there's actually a lot of interest and appropriate interest in trying to understand pre-mRNA splicing. And it's thought that the way that some of these oligos work is either by blocking or promoting RNA-protein interactions, or in some cases, blocking and promoting RNA structure.
Do you have any source of data for getting RNA dynamics?
Yeah, so right, so I alluded to that. Like that's like my pet problem. I think that's like the important problem. So I think it's very hard in any area of molecular biology to get rich information on dynamics. You can do like these beautiful NMR experiments or single molecule fluorescence experiments, but you can like typically do them like on one, like literally one kind of molecule at a time. And even if you try to increase the throughput, you can do like tens of molecules at it, tens of molecules. So that's not quite at scale. So one of the cool things that, well, this initially used to frustrate people in the RNA field, but I think it's really cool. If you take an RNA molecule, like the one I showed at the very beginning of my talk, and you create it in a test tube, and then you like add in some magnesium, which helps promote tertiary structure, and then 90% of it won't fold up correctly. Okay? Same thing is true for famous RNA machines like the ribosome. Almost any RNA you look at, you have to like then put it. There's some black magic thing where you have to put it at 50 degrees Celsius for 30 minutes and slowly cool it down, and then like 80% of the RNA that. Okay, and it was recognized early on that the RNA misfolds into alternative structures. So what its final structure is, it actually does depend on its pathway, yeah, like its history. Okay, and so historically we've then just figured out ways to heat it and cool it to try to get it into a certain minimum. But I'm damn sure that most of the pre-mRNA splicing in your body actually relies on the fact that the RNA has multiple different kinetic pathways that only occur while the RNA is being transcribed. And there's evidence for this too. So it turns out, I'll just quickly finish the your question. So we can see that too in our experiment. So the chemical mechanisms I showed you, we are heating and cooling the RNA. But if we don't heat and cool the RNA, we get a picture of the RNA as it's folded or misfolded right after it's been made. And so if you have a model that predicts RNA kinetic folding pathways, you can test it incisively in this scale, like, well, on millions and now hundreds of millions of different sequences with the same approach I just described. This is highly sensitive to structure, and the structure immediately after transcription can be highly sensitive to kinetic folding pathways. So that's like it's a super easy experiment actually, so I shouldn't call it like my dream, but I could go do it tomorrow. But I actually don't know how to make the model. I can learn from those things. How do you make a model that's predicting not just structural ensemble, but like the entire kinetic folding path? It's just a cool. I don't think it's going to be like a vanilla transformer or a a non-vanilla transformer. It's like something else.
Sort of to add to that point, you've also been alluding to
Yeah.
I guess to what extent do you think? Because I'm assuming for a lot of the experimental data, you're just IDT.
Yeah. Yeah.
So there's no sort of like RNA chaperone, THFQ, whatever else is
Right. Yeah.
assisting
Yeah.
this fold. How different do you think like the RNA structures like themselves would be like if instead you measured it like from cellular or
Yeah.
just from cell lines?
Yeah. I think it's going to be different. The question is how many? I bet you like 10% of the molecules will look pretty different. Somewhere between 10% and 50%. And like that's the same kind of thing as what I was just talking about. Like looking at RNA out of equilibrium, looking at the RNA push out of equilibrium in vivo, in E. coli, in the human cell nucleus. Like that's very doable. We have the libraries. They'll be the same damn libraries. And then that'll be the next title competition, right? Yeah, well, we don't even know the answer in vitro outside cells, except for a few special cases.
So the models that were designed taking in biological data now, like are there, what if any like physical components or physics metrics?
Yeah, so there's these classic dynamic programming methods that have been historically parameterized based on, like, actually it started here in Berkeley by he's retired, or he's passed away, Asher Tinoco. He would just take, like, here's an RNA sequence, here's a complement. What's the temperature I need to put this system at to melt the helices? So those result in so-called thermodynamic parameters are then fed into these classic dynamic programming models for modeling RNA secondary structure. And we find that if we, I mean, maybe this is obvious, but when you start to get a data set on thousands of RNA sequences and they're these kinds of data or other kinds of functional data, that we can take those same models but then derive the parameters from the data in a data-driven way, based on more data than the Tinoco lab or the other labs have collected, and those end up being more accurate for downstream tasks. So I think the RNA field is still, it's funny because 90% of the people still use these kind of old school methods, but the tide is turning.
All right, I think we'll stop there.
All right.
Thank you so much.
All right. Thanks so much.
That was awesome.
Yeah, a lot of fun. Really good questions.